Hi, welcome to Chapter 6, Enzymes. In this lecture, we're going to discuss part 1 of this chapter. Here, I'm going to introduce to you what enzymes are and why enzymes are required. These are the learning goals of this chapter. We will begin this chapter by looking at the physiological significance of enzymes. Then we will understand the origin of catalytic power of enzymes. Remember, enzymes are catalysts, and catalysts catalyze chemical reaction. Chemical mechanisms of catalysis. We will understand as to how and through what mechanisms do these enzymes catalyze reactions. Then we will look at the description of enzyme kinetics and enzyme inhibition. Finally, we will finish this chapter by looking at mechanisms of chymotrypsin and lysozyme, two different enzymes. So, what are enzymes? Enzymes catalyze a reaction. Typically, in the body, enzymes are used to catalyze chemical reactions. Now, what are enzymes made up of? Enzymes are usually globular proteins. Some enzymes are RNA molecules called as ribozymes that has catalytic properties. Study of enzymatic process is one of the oldest fields of biochemistry, and it dates back to 1700s. One of the original enzymes were extracted from yeast. This slide describes some of the common physiological enzyme functions. Enzymes act on a wide variety of substrates, but there are only limited number of actual reactions, and you can generalize these reactions as shown in table 6.3. There are 6 different types of common enzymatic reactions. Doesn't mean that these are the only different types of reactions, but these are some of the common ones, like oxidoreductases that transfer electrons, transferases that catalyze group transfer reactions, hydrolases which catalyze hydrolysis reactions, lyases responsible for cleavage of CC, CO, or CN bonds, isomerases, as the name suggests, is transfer of group within molecules to yield isomeric forms, ligases, formation of CC, CS, and CO and CN bonds by condensation reactions. Ligases are typically the opposite of lyases. Now, if you look closely, the names of the names of the enzymes all end in aces, which means that these are the names of the enzymes. If you remove the ES from the end, you get the reaction itself. For example, hydrolysis reaction is catalyzed by hydrolases. A ligation reaction is catalyzed by ligase. Isomerization reaction is catalyzed by an isomerase. Henceforth, the molecule an enzyme acts upon is called as its substrate. For example, if E were to be an enzyme, it acts upon its substrate S. So S is a substrate, and this can result in the formation of product E plus P. Substrates bind the active site of an enzyme. The result of the enzyme catalyzed reaction is the product. P, as I said, is the product and S is the substrate. And E is the catalyst. Enzymes often have help. Some enzymes can catalyze their reactions independently of other molecules, except the substrate, of course. Many enzymes require additional molecules called cofactors. Cofactors are molecules that help an enzyme in a chemical reaction. For example, inorganic ions, complex organic or metallo-organic molecules known as coenzymes, or very tightly associated or covalently bound cofactors are known as prosthetic groups. Please remember these terminologies. Cofactors act as transient carriers of functional groups. For example, cofactors can be divided into metal ions or coenzymes. Coenzymes can be further divided into co-substrates and prosthetic groups. If you remember hemoglobin and myoglobin, right? Myoglobin and hemoglobin had a cofactor. It is the heme molecule. Now, the heme molecule is attached to the enzyme or attached to the protein hemoglobin and myoglobin. They heme or a porphyrin ring is an example of a cofactor. Enzymes that work with cofactors can have two different forms. The first one is called as an apoenzyme or an apoprotein. Enzymes in the absence of their cofactors are called apoenzymes. Remember, only those enzymes that utilize cofactors have this form. When in complex with the cofactor, the complex is called a holoenzyme. Now, not all enzymes use cofactors, and these terminologies only apply to those enzymes that utilize a cofactor for catalyzing a reaction. Catalysis can be explained on the basis of energy diagrams. For example, if you consider this equation, E, which is the enzyme, plus S, which is a substrate, going to ES, which is the first enzyme substrate complex, going to EP, which is the E plus product, which then leads to enzyme plus product. Now, this is a typical enzymatic scheme. I will explain it to you in detail a little later. But here is an energy diagram which has energy on the y-axis and reaction coordinate on the x-axis. If you have not encountered this kind of a diagram, let me break it down for you. Shown on red, that is here, is the non-enzymatic reaction. Reactants are going to products. For example, in this case, this is a glucose molecule that is getting oxidized to carbon dioxide and water. Without enzyme, reactants has to pass through a transition state. And that transition state, the energy of the transition state then goes down to products, as you can see here. With the enzyme, this transition state energy decreases, and this is the new transition state. So reactants going from here to products has now a lower energy barrier to overcome in the presence of enzyme. So that's what an enzyme does. Now, if you look at the differences in energy, this is a reactant energy here, and the product energy is on here, right? Now, you can always find the differences in energy. The starting point of reactants is here. If you draw a line, the difference between the transition state and the ground state of the reactant is the activation energy without the enzyme. Now, with the enzyme, that activation energy goes down. The overall energy released during the reaction is the energy between reactants and products. So here's the reactant's ground state and here's a product ground state. Clearly, the reactant's ground state is at a higher energy level than the product, right? So that's the energy that is released upon converting reactant to a product. Enzymes alter the rate of reaction, not the equilibrium. So if you use an enzyme in this energy diagram to convert glucose to carbon dioxide, what's happening is the time that it takes to convert glucose to carbon dioxide is decreased in the presence of enzyme. That's what it means by alter the rate of the reaction. In order to get from reactants to products, there is a transition. That's what I talked to you about, transition state. The transition state often comes with a larger energetic barrier. Transition states are not intermediates. Please remember that. Intermediates are something that are stable. Transition state is not. You cannot isolate transition states, while you can isolate intermediates. There are three major classes of catalysis. The first one is acid-base catalysis, which is one of the most common catalysis type. An example is proton transfer. In this kind of catalysis, protons are used from water or from other molecules to catalyze a reaction. The second type of catalysis is covalent catalysis. In this case, a temporary covalent bond is formed between substrate and enzyme, which is then broken to form the product. The third one is metal ion or electrostatic catalysis. In this case, coordinated charged species are formed and it catalyzes redox reactions, changing the redox state of the metal. Let's look at some examples. Acid-base catalysis. In this kind of catalysis, specific amino acids in the active sites act as proton donors or acceptors. For example, you may know and remember that glue and asp are negatively charged amino acids. If they are negatively charged amino acids, that means that they are prone to accept a proton. Positively charged amino acids such as lysine and arginine already has an extra proton, so they can donate protons like this. So this is the negative charge of glue and asp. It can either donate a proton or take up a proton. Lysines or arginines have NH3+, so it has an extra proton. It can donate a proton and become RNH2, or it can accept a proton. All these depend on the pKa of these side chains. Cysteines can get deprotonated. Histidines can get deprotonated. Remember, when we talk about histidines have two nitrogens, as you can see. And at pH below 6.5, which is the pKa of this imidazole ring, right? It's always protonated. So this proton, this excess proton, can be taken up by some other molecule, and histidine can then become deprotonated. This is the deprotonated form. When it is deprotonated, there are two electrons that are on this nitrogen, as you can see here. That's the nitrogen. Serine can be deprotonated, ROH to RO minus. Although we never talked about a pKa for serine because serine hydroxyl pKa is higher than 14, and then so we do not talk about it. But in enzyme active site, serine can get deprotonated. I'll tell you as to why that can happen. Tyrosines can get deprotonated from ROH to RO minus. Waters can serve as proton donor or acceptor too. They're very sensitive to pH. Now here is an example of acid-base catalyzed reaction. This is an ester hydrolysis. Now if you look closely, B, as written B minus, could be any of these. It could be either glue in its negatively charged state. It could be RNH2 or RS minus, or a histidine, a serine, or a tyrosine. Any of these could act as B minus. Take a proton from water, right? And this water can then attack the carbonyl carbon of an ester. The carbonyl carbon is electrophilic, right? Electrophilic. Because it has a delta plus charge and it's polarized. And if it attacks there, the electrons move towards oxygen, which is electronegative. And then you get a tetrahedral species, as shown here. This tetrahedral species can then collapse because this O minus can then go back, and one of the leaving groups can leave with a proton that is attached to any of these amino acids that are shown here. So the B minus had become HB, and then it donated the proton back to the molecule and became B minus. So enzyme started with B minus, ended with B minus. So enzyme always almost comes back. But now the new product, which is the acid and the alcohol, is formed. So typical ester hydrolysis in the presence of an enzyme. That's what the reaction is. The next type of catalysis is covalent catalysis. In this case, a transient covalent bond between a substrate and an enzyme is formed, and it is necessary for the activity of the enzyme. Example, deacylation of substrate is accomplished via temporary acylation of an enzyme. Now let's look at it using an example. Here, if you look closely, the yellow arch is a cartoon representation of an enzyme. The OH group that is shown is in the active site. It's more likely to be a serine. That is the hydroxy that is shown here. What happens is a base, as shown in the previous slide, abstracts a proton, that this hydroxy, and oxygen can then attack an amide or an ester. What happens is it pushes the arrow there, and it pushes out the leaving group. This is called as an acylation reaction. And during this acylation reaction, leaving group leaves as XH. This leaves with an acyl enzyme. That is because the serine hydroxy, as shown here, is acylated. After this, water attacks, and when water attacks, a similar reaction happens. Right? Another base now removes a proton. Now it doesn't have to be another base. It could be the same base that is shown here, because this base could have then donated a proton to the leaving group X, this specific proton that is shown here. So the same base abstracts the proton from a water, and oxygen then attacks this carbonyl carbon center. And Electrons move there, and what happens is this bond is then broken, and it is taken when it breaks, it takes a proton, and the proton is taken from B minus. This results in OH, and the product acid is formed. So this is a typical example of a covalent catalysis. Metal ion catalysis often happen in presence of metals. Metal ions can help coordinate charge species, stabilizing the transition states. For example, copper 2 plus can stabilize this carboxyl and a carbonyl. And a reaction, the decarboxylation reaction, happens when like this. So a negative charge pushes the electrons inside, and the carboxyl leaves by pushing the electrons inside this bond, which then polarizes the carbonyl, and you get an enolate. This enolate can then isomerize to a ketone. Metals are good Lewis acids and effective electrophiles, and in this case, copper 2 plus acts as a Lewis acid. Alkali metals are usually loosely bound and play structural roles. Transition metals, on the other hand, usually play a functional role in catalysis as part of a functional group. So, what do enzymes do? Enzymes increase reaction rate by lowering the energy associated with the transition state. As you can see in this energy diagram, which is exactly what we saw before, the function of the enzyme is to decrease the energy associated with the transition state, and as a result it increases the rate of the reaction. That is the function of an enzyme. So the obvious question is, how do enzymes work? Or how do enzymes lower the energy barrier? Enzymes will often derive energy from substrate binding. So when a substrate is bound on an enzyme, there's always some energetic contribution that is involved, and that is the delta GB, or delta G of binding. Weak forces are energetically optimized for transition state over substrate or product. So weak forces, as we have seen before in protein function chapter, when a substrate binds to an enzyme, it binds via weak interactions. And weak interactions are energetically optimized for transition state. So it is indeed the transition state of the reaction that is binding to the enzyme and leading to a product. Let me explain this to you in much more detail. Before that, I want to get your attention to something here. Binding and specificity. What are these two terms? You know what binding is. Binding is the attachment of a substrate to an enzyme, or attachment of a ligand to a protein, right? Specificity is how specific a ligand or a substrate is to a protein or an enzyme, respectively. In other words, specificity is the ability of an enzyme to recognize its substrate. Binding energy contributes to specificity of enzymes as well as catalytic capabilities. Binding energy is derived from multiple sources. For example, weak interactions. What are those? Hydrogen bonds, hydrophobic interactions, ionic interactions, or salt bridges, and van der Waals forces. Right? Transient covalent bonds. Transient covalent bonds, the ones that we talked about for acylation reactions and stuff like that. Entropy reduction. We briefly talked about this in chapter 2. Easier to attach A to B if they are stuck into the same enzyme, right? So orientation matters. Induced fit is another concept that will be introduced to you. Induced fit allows enzymes uh, to have new molecular interactions. Let us consider a thought experiment to understand how enzymes work. Before that, let's rank order of enzyme binding to various molecules. Enzyme binds much stronger to a transition state of the reaction as compared to substrate or products. So this is a thought experiment. Let us imagine that we have an enzyme called a stickase. A stickase is the name of the enzyme, and ASE at the end always means an enzyme. A stickase, as the name suggests, breaks a metal stick. So metal stick S is the substrate, and once it breaks, the products are P. You could call P1 and P2, doesn't matter. Now what this enzyme does, it breaks metal sticks using magnetic forces. And from going to S to P, there is a transition state, and that is the bent stick. You can imagine it yourselves. If you want to break a stick, and if you want to break it with two hands, or if you want to use some gadget to break a stick, either ways, if you want to break a stick, you have to first bend it, and that is the transition state. So here is the experiment. Now there are three different scenarios here, and for each scenario on the right is the energy diagram, and what we are looking for is the delta G double dagger, and that represents the activation energy, that the transition state, the activation energy that is required to reach the transition state before it gets to product. Now if you have a metal stick, good luck bending it and breaking it, because it takes a lot of energy, right? So that's why the activation energy is so high. Now let us consider an enzyme that is complementary to a substrate. Now if it is complementary to a substrate, when enzyme binds, it's ES, right? And it goes to product. Now, does it help decreasing the energy of the transition state here? Well, if you look at the energy diagram, S going to ES. ES is when the enzyme binds the metal stick. Now, ES has a very low energy, but from going from ES to products, right, it has to bend it. That is again a very high energy demand, right? The transition state energy is very high. Doesn't matter whether the enzyme binds the stick or not, it still has to break it. So very high energy barrier even then. But let's imagine the enzyme active site looks like a transition state or a bent metal. Now in this case, the enzyme can bind S or substrate and form ES, and then the transition state. And in this case, because the shape of the enzyme active site resembles a transition state, you can easily bend it and form the products. And so in this case, the energy diagram changes. Going from ES to P is much smaller here. And also, ES is not a stable form, because if it's a stable form, it takes much more energy to go and cross the activation or cross the transition state. So ES is about the same energy as that of S in this case, in which an enzyme has a complementary the enzyme has an active site that is complementary to the transition state. From going from ES to P, the activation energy has decreased considerably. So the energy change, that is delta Gm, is the change in the energy that is brought about by an enzyme that has an active site, complementary to the transition state. So here is an example and a difference between an intermediate and a transition state. Intermediate is a chemical species with finite short lifetime. It's longer than 10 to the minus 13 seconds, but still there is a finite lifetime. Whereas transition state is maximum energy species formed on reaction coordinate. There is no defined lifetime. And the way you can explain explain the difference between an intermediate and a transition state is by pointing to an energy diagram. In this case, the top of the peak represent transition states, and the bottom or the peak that is or the space between the two peaks represents an intermediate. Now, remember, this is neither the substrate nor the product. The substrate starts here, product ends here, and it is between the substrate and product, but has a finite short lifetime. That's the difference between an intermediate and a transition state. So the biggest takeaway from this lecture is that enzyme has complementarity to transition state of the reaction. When the enzyme binds the substrate, there are some weak bonds, but when it binds the transition state of the reaction, it has many weak bonds that contributes favorably to decrease in the energy of the transition state, leading to formation of products. You can also watch this video for studying purposes. It's really helpful to understand it much better.